All right, hello biology students. The purpose of this video is to outline what you need to know for B 2.2 and B 2.3. So this is all about cells and their form, so their structures and how those structures help to support their functions. And so we have two sections we're going over here. B 2.2 is all about organelles. And how they're adapted for specific functions, and what the advantages are to having these organelles or different compartments within cells. B 2.3 is really about two things, sort of two extremes of cells, very undifferentiated cells called stem cells, and then specialized cells, which we sometimes call differentiated cells, and how those work, why they're important, and how one becomes the other. So we'll be going over all of these in this outline. We're going to start here with organelles and how they're adapted to their functions. So what I'm going to do in this video is always start with the curriculum statement, which comes from the IB. It sort of like helps to focus our attention on what we have to know. And then we'll go from there. So starting from the beginning, B 2.2.1, it's basically defining what an organelle is. So organelles as discrete subunits of cells that are adapted to perform specific functions. Discrete meaning you can identify them clearly. Certain things are organelles and certain things are not, and we need to know what those are. If you look on the bottom there, this thing that says NOS, that second bullet point, that means nature of science. These parts of the curriculum, they will show up on exams in a small way. You might also find they will link quite well to TOK. So pay attention to those for those two reasons. So just starting out here, what is an organelle? This is a nice diagram showing many different cell types. I like this because it shows the variety of cell types. So the big oval cell in the middle there, I think that's a pancreatic cell. A pancreas is an organ in your body that produces insulin. And then we see around there on the top and the bottom, there are capillaries, which are small blood vessels, and you can see there are these thin cells lining the capillaries called endothelial cells. And on the bottom, there's another kind of cell, a neuron, and the yellow cell, or at least a part of a neuron. It's labeled there, cholinergic nerve ending. Okay, so you do not need to know all this stuff right now, but I'm showing this just to give you a sense of the diversity of cells. And on the big one in the middle, we can see a lot of organelles in there. We can see a nucleus, endoplasmic reticulum, mitochondria, Golgi apparatus, and vesicles as well. And we can start to understand what those are for, their functions, and how they're related to each other. One thing I want to point out for this topic is very much an overview of several different things related to cells and organelles. And the idea is to just touch on each of them as a little bit of an introduction. And for a lot of these topics, we're going to come back to them later and go into them in way more detail. So it's always the case when we're starting out in biology, you probably feel like you need to study everything at once. I understand that. Try to resist that urge a little bit. Just try to take this in as an introduction and trust that we will come back to many of these topics later on. Okay, starting very basic, you should be able to identify which of these structures are organelles and which are not. And so there it is nicely organized for you. You can see the cell wall, cytoskeleton, and cytoplasm are not considered organelles. Cytoskeleton and cytoplasm are not discrete structures. They're just sort of spread out within a cell. So they're not considered organelles. And the cell wall is actually outside the cell. It's part of the extracellular, it's part of the extracellular portion of the cell, like the outer part. And so we don't consider it an organelle either. The other structures are organelles. I think it is useful to have like a one-sentence summary of each of their functions. I think it's also useful to know how many membranes surround each of these organelles. And so here it is nicely outlined for you. So ribosomes and centrioles, they are considered organelles, but they have no membrane. They are not membrane-bound organelles. That is a little bit important. Most organelles have a single membrane around it. When I say membrane, it's a phospholipid bilayer, just like the plasma membrane. So vesicles, vacuoles, rough and smooth ER, Golgi apparatus lysosomes are all single membrane organelles. And you should really know which ones are double membrane. Those include the nucleus, or plural nuclei, mitochondria, and chloroplast. They all have a double membrane. All right, you might wonder, how do we know all the stuff we know about organelles? If you've looked at a cell under our light microscopes, you can't actually see very much. You can see a nucleus, you can see chloroplasts in a plant cell, cell walls. That's sort of about it. So this slide is related to one of those NOS, Nature of Science, curriculum statements, which also links nicely to TOK. And specifically, this statement is saying that progress in science often follows the development of new techniques. So techniques like microscopy, there's all different kinds of microscopes. There's different techniques as well in microscopy. We have electron microscopes. We have techniques involving fluorescence. That's where this image on your bottom left is from. We will look at some different kinds of techniques in microscopy later, but just be aware, technology that we don't have in the school has led to developments in our understanding. And there's other techniques too. So on your right, you can see this these techniques of using a centrifuge, which spins things around really quickly and can separate them by mass. It's called cell fractionation. And you can separate organelles within cells in this way. So you can see here, there's a way to get out the mitochondria and the chloroplasts and the ribosomes and so on, just by spinning or centrifuging mass of cells around. So we know about organelles based on the technology that we have to see them and to understand them. Okay, moving on to B 2.2.2. We're now getting into discussing specific advantages of compartmentalization in different organelles. Like I said before, a lot of this is just introducing topics that we're going to get into in a lot more detail later. So there's going to be new vocabulary. You might feel like there's too much information, but again, trust that we will come back to a lot of these topics. So the first one is, what are the advantages of separating the cytoplasm from the nucleus, from the contents of the nucleus? And you can read there's a few key points you should focus on. One is separating gene transcription from post-transcriptional modification of mRNA. And we haven't studied protein synthesis yet. So that might not be familiar with you, but that's okay. Let's look at that a little bit though. So here's a nucleus. You can see the nucleus is one of those organelles with a double membrane. And the membrane has these pores in it. These are large pores, larger than the kinds of openings you would have in a protein, like an integral transport protein in a plasma membrane. These pores create much larger openings. The image on your right is an electron micrograph of a nucleus. It was made using a technique called freeze fracture. So basically a cell is frozen and then fractured, and it breaks along areas of weakness. So you can actually see you can see part of the double membrane has been split in some regions. You can see the sort of jagged edge. So that provides evidence that it is a double membrane, and you can also see the pores. So it's really good example of how our understanding of these structures has developed through the use of technology. Okay, but I haven't answered the question. Why do we separate the nucleus from the cytoplasm? One is DNA is incredibly important, so it makes sense to have it in its own compartment. where it's not at risk of being damaged by all the chemistry happening within the cytoplasm. Another useful feature of separating the DNA is it gives us a little more, it gives cells a little bit more control. So remember, DNA is used as a recipe to create proteins. This process is called protein synthesis. Protein synthesis involves two steps: transcription, which is copying the DNA instructions. That copy is called messenger RNA or mRNA. And then a second step called translation, which is then reading those instructions, reading the mRNA, and making proteins from that mRNA. So transcription happens in the nucleus, in cells with a nucleus, eukaryotic cells which have a nucleus. And translation happens in the cytoplasm, specifically at the ribosomes. And so at the ribosomes, the mRNA attaches to them, and the mRNA is then read by those ribosomes, and amino acids are brought in the correct order to make polypeptides, which then form proteins. Now, I just referred to this briefly, but we can roughly divide cell types into two: prokaryotic and eukaryotic. So eukaryotic cells, the one on your right there, are like our cells. All animals and plants are made of eukaryotic cells. Not only animals and plants, but certainly animals and plants. And eukaryotic cells have membrane-bound organelles. They are compartmentalized. Prokaryotic cells do not have membrane-bound organelles. They do have some organelles, they have ribosomes, but they don't have these membrane-bound organelles creating different compartments. So if you look on your left, you can see that the processes of transcription and translation happen at the same time. As the DNA is copied to make the mRNA, it is at the same time transcribed to make proteins. So that does not allow any possibility to regulate or modify the mRNA, the copy that's made, before it is used. Okay, it has to be used right away. In a eukaryotic cell, on your right there, so the purple region is the nucleus, and the area outside of that is the cytoplasm. You can see, if you look carefully, the mRNA is formed in the nucleus, so that's a copy of the DNA. And then it has to leave the nucleus through one of those nuclear pores, where it is then used to make a protein at the ribosomes. Because there is this separation between the nucleus and the cytoplasm, there's an opportunity to modify the mRNA, and that's what happens. That's called post-transcriptional modification. Post means after, so after transcription, modification changes. So if you think of mRNA as a recipe, basically the recipe can be adjusted a little bit before it's used, which is useful. It allows for more flexibility in how the genetic code is used to make proteins. Okay, going on to the advantages of compartmentalization in general. So again, this is something all eukaryotic cells do because they have membrane-bound organelles. And just in general, why is it advantageous to separate or to create these different compartments? And you can see there in the curriculum statement what you need to focus on. This idea of concentration, the idea of separation, so that we can separate things that don't go well together. And you should be able to talk about lysosomes and phagocytic vacuoles as specific examples. Okay, so let's go through this a little more carefully by looking at an example, which is garlic. Garlic is a plant. It's made of cells. And if you do any cooking, you know that when you crush garlic or slice it, it creates a smell, right? And we like that smell and that taste in our food. But if it's uncrushed, it doesn't really smell like anything. So why is that? The reason for that is the enzyme that is required to create the chemical that creates the characteristic smell of garlic is compartmentalized. It's separated in an vacuole within the garlic cells. When you crush garlic or you slice it, you actually break that vacuole and you release this enzyme. It's called allanase, and that enzyme causes a chemical reaction that takes a metabolite called L-alanine and it changes it. It reacts with it and changes it to allicin, and then allicin is the compound that creates the odor. We like it in our food. For the garlic, it's actually a way to protect itself. It's like a chemical defense mechanism. So there's some vocab there that you may or may not be familiar with. So enzymes, these are proteins that catalyze chemical reactions within cells. Metabolites are the chemicals that are either the reactants or the products of all of these chemical reactions within cells. So you have to understand that within a cell, for that cell to function, there are thousands, perhaps more chemical reactions happening, and they all need their own particular conditions, and by creating separation, compartmentalization, we can ensure that those reactions can happen separately and have some control over them. And actually this first bullet point is also about concentration. So if you have a certain metabolite or enzyme within a enclosed space, within a vacuole, you can create a higher concentration than if it's just spread out everywhere within the cytoplasm, and that allows it to be more effective and more efficient. And some enzymes would damage the cell if they were just free within the cytoplasm. Digestive enzymes are a good example. So by creating this compartmentalization, by putting these enzymes in a vacuole, for example, we protect the cell from that damage. And finally, we can, within different compartments, we can have different conditions, for example, different pHs that are suitable for whatever enzyme, whatever chemical reaction is occurring. So it can create, again, a level of control and specialization for different chemical reactions within different parts of the cell. Here's an example you need to know about, specifically the vocabulary lysosome and phagocytic vacuole. So looking at this diagram, this is a cell, and we see endocytosis happening. This cell is an immune system cell. It's a macrophage, sometimes called a phagocytic leukocyte. We will study the immune system later. So this is one of those things like we're just introducing it. Don't get too caught up in the details. But these kinds of cells, they are constantly monitoring, and if they encounter a pathogen, in this case those are microbes, kind of bacteria, they will take it into their cell through endocytosis, and they'll form what's called a phagocytic vacuole. Here it's just labeled vacuole in that diagram. And then, so that vacuole now contains the pathogen, and that vacuole will then fuse with a lysosome containing digestive enzymes. Then the enzymes will come into contact with the pathogen and break it down, and that pathogen is then destroyed. Okay, next up we're going to look at mitochondria and chloroplasts, and look at the structures in mitochondria and chloroplasts that help facilitate cell respiration, which happens in mitochondria, and photosynthesis, which happens in chloroplasts. Now, this section is difficult to understand at this point in time because we haven't studied cell respiration and photosynthesis yet. We will study it later. So it's going to feel like there's a lot of information here. Again, try to just take it in as an introduction and trust that we will get into it in more detail later. So here's an electron micrograph of a mitochondrion, image taken using an electron microscope. And we can see some of the structures you need to be aware of. It's a double membrane around a mitochondrion. There's an outer membrane and an inner membrane. And the inner membrane is folded into these long folds called cristae. The space between the outer and inner membrane is called the intermembrane space. And the space within the two membranes, the fluid-filled region within the two membranes, like inside the mitochondrion, is called the matrix. You need to remember all of this vocabulary. And so here's where understanding how those structures are supporting the function, we do need to consider the function of cell respiration a little bit. We have to be careful here because this is a huge detailed section that we're going to study later. So I'm just going to touch on some of these things, but you don't really need to know all these details. Certainly you don't need to know all the details in this diagram yet. But just to orientate you a little bit, the yellow structure in that diagram, it's labeled there, is the mitochondrion. And you can see the double membrane, and you can see the infolding, right? And the different processes shown there are the different processes of cell respiration. Now, cell respiration actually starts outside the mitochondrion in the cytoplasm. You can see glycolysis there is the process. Again, you don't need to know the name yet. But the products of that first step of cell respiration pass through the two membranes of the mitochondria and enter the mitochondrion. And within the mitochondrion, in the inside, in the matrix, so you should be aware of that region now, the matrix, there are all sorts of enzymes to facilitate the citric acid cycle. So that's part of cell respiration. It's sometimes called the Krebs cycle. And so the matrix is an environment where we have all the enzymes necessary for that to occur. Another big step is this thing called oxidative phosphorylation, which is the process that makes most of the ATP in cell respiration. Remember, ATP is the energy molecule that cells all use as their energy source for things like active transport, and it's made through cell respiration. Now, to make ATP, we need various proteins that are in the inner mitochondrial membrane. So the reason the inner mitochondrial membrane is all folded into these cristae is to create more surface area so we can have more oxidative phosphorylation and make more ATP more efficiently. Another critical structure for oxidative phosphorylation is the intermembrane space, the space between the inner and outer mitochondrial membrane. That space has a very small volume, like compared to the matrix. And so that's really important because it allows for a concentration to build up rapidly. And I'll explain this more in a minute. But it's a concentration of hydrogen ions or protons that build up within that intermembrane space. And that is a necessary part of oxidative phosphorylation. So as a result of that small volume in the intermembrane space, we can make ATP more efficiently. So here is a summary of all those various structures that you should be aware of. The inner membrane there is folded into cristae. So if you look on the cristae box there, it says it increases the surface area to volume ratio. It means we get more surface area per unit volume for the oxidative phosphorylation portion of cell respiration to occur. The outer membrane, you can see it has transport proteins, so it can bring in what's called pyruvate, which is the product of the first step of cell respiration, which happens in the cytoplasm, into the matrix. And the matrix has all the enzymes and the correct pH for those enzymes to work, to function, making the Krebs cycle, also called the citric acid cycle, possible. And lastly, that intermembrane space is small in volume, which allows protons or hydrogen ions, same thing, to accumulate quickly, making them more effective. Okay, I know that was a lot of information. We're going to come back to it in this video again after I talk about chloroplasts, but we're going to come back to it in the future in way more detail. So looking at the chloroplasts now and how they are adapted for photosynthesis. Here's an electron micrograph of a chloroplast. So similar to the mitochondria, you need to be familiar and remember some of the structures. So instead of what we call the matrix in the mitochondria, we have the stroma in the chloroplast. That's the fluid-filled region within the chloroplast where there are enzymes and a suitable pH for chemical reactions to happen. In this case, it's what's called the Calvin cycle. We have a double membrane here as well, an inner and outer membrane. They're a little less significant than in the mitochondria. The inner membrane is not folded. But instead of having an infolding of the inner membrane, we have these other membranous structures. Here they're labeled, you can see the granum labeled. Many granums are actually called grana. And they are these stacks of membranous structures that have a large surface area. So in a sense, they're analogous to the cristae of the mitochondria because they increase surface area for a certain stage of photosynthesis to happen. So again, just like with the mitochondria and cell respiration, we do need to be aware of some of the processes of photosynthesis to make sense of the structures, to understand how the structures are supporting the function. So here I am inside a chloroplast, and you can see photosynthesis consists of two stages. The light reactions, as they're labeled here, sometimes called the light-dependent reactions because they require light. Those happen inside the grana. I'm going to add a new vocab here. Grana are made of these stacks of thylakoids, and the thylakoids have a large surface area to make that process more efficient. And then the other main process in photosynthesis is the Calvin cycle, sometimes called the light independent reactions because it doesn't need light. And that happens in the stroma with the help of a lot of enzymes and the right conditions which exist in the stroma. And here we have a nice summary of the key structures. So the stroma there at the top, labeled there, has the right enzymes and the right pH for the Calvin cycle to occur. The thylakoids, you can see it there labeled. A stack of thylakoids is a granum. Many granums are grana, plural. And the grana, because they're these thin stacks of membranes, create a large surface area to volume ratio, which allows the light-dependent reactions to happen more efficiently. The grana, by the way, are where the chlorophyll is. The chlorophyll is what makes plants green and absorbs light and makes photosynthesis possible. Another adaptation of the grana, the thylakoids specifically, is they have a very small volume within them. This is analogous to the small volume within the intermembrane space in the mitochondria. It allows hydrogen ions, protons, to build up rapidly, which is necessary for the light dependent reactions to occur. The other structures there, the lamella, don't worry about those. Double membrane, you should know they exist. You don't really need to think about the significance yet. Okay, what's this slide about? So I want to go into a little bit more detail on cell respiration and photosynthesis, just so this makes a little bit more sense. But just know this is actually not part of this topic. So why am I saying building shelves to fill with books later? A lot of what we're doing in biology now is like creating a place, an outline, which we're going to fill up with the details, the books, later on. So if you can think of it that way, I think that's a nice analogy. We're making bookshelves. We'll put all the information on it later. But a little bit of information now is maybe useful. So I like this image here because it's showing an analogous process in the inner mitochondrial membrane during cell respiration and in the thylakoid membrane during the light-dependent reactions of photosynthesis. And so these are all kinds of what we call phosphorylation. In mitochondria, in cell respiration, we need oxygen to make this happen, so that's why it's called oxidative phosphorylation. And in the chloroplasts during photosynthesis, we need light. That's why it's called photophosphorylation. And what in the world is phosphorylation? Phosphorylation is the process of making ATP. Specifically, it's combining ADP and inorganic phosphate, PI, to make ATP. And in the mitochondria and chloroplasts, we do this process. To make ATP, what we need to do is make a hydrogen ion or proton gradient. And this is where this small volume thing comes into play. So you can see beneath me in that box there, there's a membrane and there's a hydrogen ion being pumped into a certain space. So in the mitochondria, that space is the intermembrane space. In the chloroplast, that space is the space inside the thylakoids, called the thylakoid space. And both of those spaces have a small volume, so we can build up very rapidly a high concentration of protons within that space. That's the adaptation. Those hydrogen ions are then used to create ATP in this way. So what happens is the hydrogen ions will flow down through a special protein called ATP synthase. It's sort of like similar to kind of facilitated diffusion. So the hydrogen ions are going down their concentration gradient after they've been pumped first into that space. And then the energy of that flow of hydrogen ions is used by the protein ATP synthase to assemble ADP and inorganic phosphate into ATP. That's phosphorylation. It happens in the mitochondria and in the chloroplasts. It depends on a small volume within the intermembrane space in the mitochondria and a small volume within the thylakoids in the chloroplasts. Here you can see these processes in more detail. So on your left is oxidative phosphorylation happening in the mitochondria, and on your right is photophosphorylation happening in the chloroplasts. And in both cases, you can see hydrogen ions are being used to drive the production of ATP. Phosphorylation in both cases involves two steps. One is called an electron transport chain. Don't worry about that yet, but that's what's used to create the proton gradient. And then this chemiosmosis is the proton gradient being used to make ATP by ATP synthase. In the thylakoid, these protein complexes called photosystems are where the chlorophyll is, and they collect the light. So by having this large surface here, we can collect a lot of light. So you should be able to answer that question on the bottom there based on my explanation and these diagrams. Important point there, the last bullet point. In cell respiration, oxidative phosphorylation is how we make the ATP that we're trying to make. In photosynthesis, we're making ATP in this step, but that ATP is actually used in the next step in the Calvin cycle as an energy source to make glucose. Remember, the goal of photosynthesis is to make glucose. So the ATP is just an intermediate molecule that's used up again. We need it, but it's used to make glucose. In cell respiration, ATP is the product. That's what we're trying to make. Just to point out what the Krebs cycle and the Calvin cycle are. They're these very complex enzyme catalyzed reactions. They're cycles. So one chemical, one metabolite reacting to produce another, and each step is catalyzed by a chemical reaction and requires very particular conditions that for the citric acid cycle or the Krebs cycle exist in the matrix of the mitochondria, and for the Calvin cycle or the light independent reactions exist within the stroma. We will look at these later on. You do not need to know all the steps right now. Here's a nice slide with some vocabulary. I've thrown a lot of vocabulary at you. I am aware of that. I know it's difficult to take this all in, but maybe spend a few moments reading through these and focusing on those bold vocabulary. So the top bullet point is about mitochondria and cell respiration, what's happening there. And the bottom bullet point is about the chloroplasts and photosynthesis and what's happening there. And I've taken all the key vocab of the different processes and highlighted them for you. Okay, one more summary slide because that was a ton of information. So what you need to know, taking a step back from all the detail, is the mitochondria has the matrix, which compartmentalizes the enzymes and substrates, the metabolites, the chemicals involved, in the Krebs cycle. The inner mitochondrial membrane has a large surface area, so we can maximize this process of oxidative phosphorylation. And the small intermembrane space allows protons to accumulate rapidly, which again makes oxidative phosphorylation, this process that makes ATP, possible. In the chloroplasts, we have analogous structures. So the stroma is what's compartmentalizing enzymes and substrates, in this case for the Calvin cycle, sometimes called the light-independent reactions. The flattened thylakoids, which form the grana, have a large surface area, which includes all these photosystems which collect light, maximizing this process of photophosphorylation. And the small thylakoid space allows rapid accumulation of protons required for photophosphorylation, which makes ATP, which is required to make the Calvin cycle possible. All right, that was a lot of information, and we're still going. We're moving on. Hopefully the next few topics here are a little less dense than that last one. So moving on to the nucleus and the function of its double membrane. All right, so why do nuclei have two membranes? So looking at this, first just notice that there are two phospholipid bilayers. So in other words, 4 layers of phospholipids around the nucleus. That's quite different than the plasma membrane, which has 1 phospholipid bilayer, or 2 layers of phospholipids. So yeah, there's an inner and outer membrane. Why is that the case? One reason is that having 2 membranes allows for larger pores. So if you just have a single membrane, the pore size is limited to the kind of opening that you could make in a single, like a transport protein. But when you have a double membrane, you can create a larger pore size, and that allows larger things to go in and out of the nucleus, which the nucleus needs to do. So the nucleus needs to send messenger RNA out, send ribosomes, which are assembled within the nucleus, out, and bring proteins in that are necessary for transcription and post-transcriptional modification in the nucleus. So big things need to go in and out. So therefore, large openings or pores are required, and those are only possible with a double membrane structure. The other reason they have 2 nuclei is to facilitate mitosis and meiosis. Mitosis and meiosis are cell division, and during cell division, the nuclear membrane has to break down so that the DNA, the chromosomes, the genetic material, within the nucleus can be divided up into daughter cells. And so a double membrane allows that nuclear membrane to break down. When it breaks down, it actually forms these vesicles, and the double membrane structure makes that easier to happen. Okay, moving on to ribosomes. So ribosomes are the structures that do protein, or do part of protein synthesis. They do the translation phase of protein synthesis. They are the site where mRNA is read, is used. mRNA is used to put amino acids together in the right sequence. And so at the ribosomes, amino acids come in and they're assembled, they're put together, peptide bonds are formed between them, forming polypeptides, which ultimately form proteins. Okay, so we need to talk a little bit about endoplasmic reticulum. There's two kinds of endoplasmic reticulum. We abbreviate it ER. There's rough and there's smooth. They're interconnected to each other. The smooth tends to be more like tubular in structure, whereas the rough tends to be more like these flattened kind of membranous sacs in structure. And the rough is called rough because it has ribosomes bound to it. So here's a nice little Venn diagram showing you the similarities and differences between rough and smooth ER, just the main ones in terms of their function. So main one is rough ER is covered in ribosomes, smooth is not. But they both make materials for the cell. That's pretty general. Rough ER, since it's covered in ribosomes, is making proteins, whereas smooth ER has no ribosomes. It's not making proteins, but it'll make lipids, like phospholipids, and steroids as well, which are a kind of lipid. So looking at the ribosome in a little more detail, ribosome's made of these two subunits, large and small. They're made of a mix of what we call RNA and proteins. And what they do is they basically attach to the messenger RNA. Remember, that's a copy of the DNA. The messenger RNA sandwiches between the large and small subunit. And then they have these sites where tRNA, transfer RNA, can come in, carrying amino acids in the correct sequence based on the code in the mRNA. So the mRNA is a genetic code, it's a recipe that's used by the ribosomes to put amino acids together in the right order. I basically just summarized protein synthesis or translation very, very quickly. Again, this is another topic, just like cell respiration and photosynthesis. That's a complicated topic that we'll come back to again later. There's another nice diagram of a ribosome showing the different binding sites and where the mRNA binds and where the polypeptide forms. Okay. So we talked about the ribosomes on the rough ER. Those are called bound ribosomes. We also have ribosomes just floating around in the cytoplasm. They're called free ribosomes. Both of them make proteins. They have the same function, but the outcome of the proteins made at bound and free ribosomes is different. So free ribosomes make proteins that are released into the cytoplasm and used within the cell, either in the cytoplasm or sometimes in the nucleus. They'll go through those pores into the nucleus. So yeah, free ribosomes make proteins used in the cell, whereas bound ribosomes, again, the ones attached to the rough ER, make proteins that are transported somewhere. Often transported to the Golgi apparatus and then ultimately secreted out of the cell. That's often the case. Sometimes they're transported to the Golgi apparatus and put into a lysosome, like digestive enzymes. So the enzymes in a lysosome would be created at ribosomes bound to the rough ER. So the Golgi apparatus is working together with the rough ER very closely, and it's responsible for processing proteins that will be secreted, usually secreted from the cell. And here's a nice diagram showing the interconnections between the nucleus, the rough and smooth ER, and the Golgi apparatus, and the vesicles that form in those different locations. One thing that it's not clear in this diagram, but one thing to note is the rough and smooth ER are part of the nuclear membrane. Like the nuclear membrane can be attached to rough and smooth ER. They're all made of phospholipids, so they can be continuous with each other. So just a small thing to note there. When proteins are made at the rough ER, they'll be packaged into little transport vesicles. You can see there in yellow, those vesicles will then fuse with the Golgi apparatus. The proteins go into the Golgi where they're modified. The Golgi then makes new vesicles. In this case, you can see it's made secretory vesicles, which will then fuse with the plasma membrane. Exocytosis will happen, and the products are released from the cell. And so, yeah, Golgi processes polypeptides. Just a few structural notes about the Golgi. There's two sides. There's the side that receives vesicles from the rough ER. That's called the cis side. And there's the side that where vesicles form after the whatever protein has been processed and is ready to be released, and that's called the trans side. Either released into a lysosome or into a vesicle that's going to be bound or traveling to the plasma membrane for exocytosis. There's a nice electron microscope image of a Golgi apparatus. They always look like this, several layers of flattened sacs with lots of vesicles around it. They're quite compact. Some students mix them up with endoplasmic reticulum. So you should be familiar with what these things look like. Just a couple examples of how proteins can be processed, and this links to the previous topic about proteins. So glycoproteins are created in the Golgi apparatus. The carbohydrate part that's added to a protein to make a glycoprotein, that happens in the Golgi. Also, polypeptides can be assembled to make the quaternary structure of a larger protein. That often happens in the Golgi apparatus as well. Okay, I think we're nearing the end of B 2.2, looking at structure and function of vesicles. And there I am in a vesicle. And this is a vesicle that looks like it's going to fuse with the plasma membrane. Yeah, it's fusing with the plasma membrane. And then the contents of that vesicle, me in this case, is being secreted from the cell. And so what is a vesicle? It's a phospholipid bilayer. It's made of the same thing as the plasma membrane, in fact. It can be made from the plasma membrane. It can be made from the Golgi. It can be made from the ER. It's all the same stuff. So vesicles can be made within the cell and fuse with the plasma membrane to release substances outside of the cell. That's what's being shown here. Or things can be taken into the cell, sort of the reverse of what's being shown here, and a vesicle can form from the plasma membrane. When things come into a cell and the plasma membrane has to fold inwards, that takes a little bit of energy, and it's not necessarily an easy thing to do because the plasma membrane wants to stay in its sort of like the easiest, the lowest energy state it can be in, and pushing it in is sort of like stretching a rubber band. It takes some effort. So there is a protein you need to be aware of that supports that effort. It's called clathrin. You can see it in the top diagram nicely there. It's a three-legged protein, so it's like it has these three arms. And the clathrin proteins will bind to each other and create like this lattice or this scaffold that helps to support and indent the plasma membrane and form this sphere, this vesicle, when substances come into the cell by endocytosis. It's not the only thing required. There are other substances, like here you can see in the top one, a protein called dynamin. You do not need to know about those, but you do need to know about the clathrin. All right, we are done with B 2.2. So you might want to take a break at this time. And next we're going to move on to B 2.3. And B 2.3 is really divided into two parts. One is about stem cells. That's the first part. And the next part is about differentiated cells. So stem cells are unspecialized. And when an organism, like let's talk about a human, first comes into existence. We're just one cell. And that cell then divides to become all the cells that we end up being composed of. An adult human is like roughly 50 trillion cells. And those cells in an adult are mostly very specialized. Like you have cells for your eyes, for your nose, for your skin, for your various muscles, and so on. Immune system cells, blood cells, cells of your lungs, and so on. The point being, all of those cells are very specialized. They're very different in structure and in their function, but they all arose from the same first cell that was completely unspecialized. That first cell, that unspecialized cell, is called a stem cell. So we went from this unspecialized stem cell to highly specialized cells through this process of differentiation as that first cell divides into many cells. So when did life start? When did your life start? Looking at this diagram, you can see in your bottom left, there is an egg cell, the big red one, and there are all these sperm cells around it trying to get in there. One of them gets in, right? And the nuclei of the sperm and the egg, they fuse. And then you get this first cell that I was talking about. Is that when life starts? Perhaps there's argument about that. I would say that's not, that first cell certainly has no consciousness yet, but it is alive. And then it starts to divide. Okay, it starts to divide and divide and divide, and then we get this differentiation to form at first an infant, which then develops into an adult. And so here that is shown a little bit more clearly. The egg and the sperm, those are called gametes. After they fuse, you get a zygote. So there's that zygote. The zygote, that's that first cell. Right, that starts dividing. Once a zygote starts dividing, we call it an embryo. There are various stages of embryo. And then the embryo the cells will start to specialize, to differentiate, to form a fetus. A fetus is before birth. And then the fetus is born, and we have an infant that's going to develop into an adult. Which of these cells is specialized, which are unspecialized? So interestingly, the sperm and egg are highly specialized. Okay, they're like, think of a sperm cell. It has a tail, it can swim. An egg cell is very large. They're some of the largest cells in a human. You can actually see them. They're like about the size of a period in a sentence, maybe a little smaller. And so they're highly specialized cells. But once they fuse and become a zygote, that is a completely unspecialized cell. There we go. So that process is called fertilization, sperm fusing with egg. We get a zygote, we get this unspecialized cell. It's called a diploid cell because it has two sets of chromosomes. The sperm and the egg are called haploid cells because they have half the number of chromosomes. Again, you don't need to know all of this at this point. Just a little introduction. That zygote starts dividing and dividing and dividing and differentiating, which means the cells start to specialize. Very specialized cells. Now, the interesting thing is that zygote, the genetic information in the zygote, the chromosomes, the genes, the DNA, is copied every time the cell divides. And so in an infant and even an adult, like our 50 trillion cells, they all have exactly the same DNA, the same genetic information. So if DNA is what's used to make proteins, and the proteins are what determine what the cell is like, how is it that different cells can have different appearances, structures, and functions if they all have the same genetic information? So let's consider that. During development, so after we get that zygote forming, we start to see differentiation almost like right away, even at the 2-cell stage, which is like 24 hours later. And so the reason for that is because of these asymmetries in certain chemicals called morphogens. Okay, so what's an asymmetry? Asymmetry means these things are not at the same concentration everywhere. So they're spread unevenly across the embryo. It starts from the very beginning when the zygote forms. The point where the sperm enters, calcium ions are released there. And so there's going to be a higher concentration of calcium ions on one side of the zygote than the other. And then when that cell divides, one of those daughter cells will have more calcium than the other. The calcium ions are an example of a morphogen, and morphogens impact gene expression. So this is a long way of getting to the answer to my question, is how do cells differentiate when they have the same genetic information? It's because they use some of the genes and not all of them. So when a cell differentiates, it turns off some genes and turns some on. That's called regulating gene expression, determining which genes are on and off. And the way that's done is through these chemicals called morphogens, and the concentration of morphogens varies across an embryo because of these asymmetries that have been set up right from the very beginning when sperm enters egg. And then you get this like cascade, it's like a chain reaction where that very small difference at the beginning leads to another difference and another difference and another difference. And you get these gradients of these morphogens in different cells across the embryo as it develops. And those different concentrations of morphogens lead to differences in gene expression, and then different genes being turned on and off, and therefore different kinds of specialization. So cells can become muscle cells or brain cells or whatever they need to become. Here that is written out for you in some words. So there's the term gene expression. So again, all cells have the same genes, but the genes that are expressed vary. And morphogens are what impact gene expression. So you could call them gene expression signal molecules. And they're found in these gradients of different concentrations, with the higher concentration at the source of that morphogen, and then a lower concentration as you get further away from the source. That creates asymmetries right from the beginning in the zygote. And those asymmetries become more and more obvious, leading to more and more differences in gene expression in different parts of the embryo and as it develops. Okay, let's just take a step back to basic definitions. So we've said a stem cell is this very undifferentiated cell. A zygote is an example. What else can we say about stem cells? There's two things we can say. One is they have the ability to divide indefinitely. So they divide, making two stem cells, which then can divide again, and that can happen forever. So they have this ability to divide indefinitely, this self-renewal property. The other one is they have the capacity to differentiate into any cell type. So they can change from a stem cell to a differentiated cell, and the kind of differentiated cell they become will depend on the gene expression that is triggered by a given morphogen. We know that a zygote is a stem cell. Early embryos have many stem cells that can become many different cell types, but we still have stem cells as adults. And those stem cells live in these areas called stem cell niches in adult humans. So a stem cell niche is an area of high concentration of stem cells in an organism. And these regions provide the conditions to maintain those cells, and in some cases to promote division of them and possibly differentiation of them. So I think you need a concrete example. Two examples are bone marrow. So bone marrow is inside your bones, and that's a location where stem cells exist. And those stem cells are actually what are used to form blood cells, red blood cells. So the bone marrow stem cells will differentiate to form blood cells. And blood cells have a limited lifespan, so we always need this supply of stem cells in our bone marrow to make new blood cells. Hair follicle stem cells are in our skin, and they have the potential to produce hair as well as epidermis, like more skin, and sebaceous glands, which are like sweat, for example, producing glands, oil producing glands within our skin. So the various skin cell types. If you were looking very closely at the last slide, you might have saw the word multipotent. So that's a kind of stem cell. There are three kinds of stem cells you need to be aware of. So looking in this diagram, you can see the three kinds going from the least specialized to a somewhat specialized stem cell. So the stem cells in that, like the zygote in the very early embryo, are called totipotent. They can become any cell type. Cells then start to specialize, but they don't become completely specialized. So the next level after totipotent is pluripotent, and then after that we have multipotent. So a little bit more detail in the definition here. Totipotent can become anything. They form the complete organism. The 50 trillion cells of an adult all come from those totipotent stem cells. There's only very few of them, and they only exist very, very early in the embryo. After the zygote starts dividing, we still have stem cells, but they're now called pluripotent. They can become many different kinds of cells, almost every kind, but you could not take one of those cells and make a new organism from it. It can't become every cell type. Multipotent stem cells exist within adults, and they can become more than one kind of cell, but it's a limited number. So like the stem cells in our bone marrow, in that stem cell niche, can become different kinds of blood cells. I think I said red blood cells, but they can become different kinds of blood cells. Or the cells in our skin, what was it, root hairs? Hair roots? I think I'm getting that wrong. But that niche can form the different kinds of skin cells, but not other kinds of cells, so they're multipotent. You don't need to know about iPS cells, but I wanted to point it out because we live near Kyoto University, where iPS cells were invented by Yamanaka-san, the doctor there, and he got the Nobel Prize for that. So he found a way to genetically modify differentiated cells, like adult cells, and turn them back into stem cells. And now they're looking at applications of that, how we can use custom-made stem cells to treat and cure diseases. So it's a very exciting area of research. So those are iPS cells, induced pluripotent stem cells. So they're very unspecialized, not totipotent, but they're pluripotent, but they are induced, so they are created by genetic modification. Okay, we're almost there. So we're coming down now to the second part of B2.3, which is about differentiated cells specifically, and just running through several examples of how the sort of diversity of cells that exist we're looking at humans mostly here, and how they're specialized to specific functions. All right, the first thing to talk about is cell size. So one aspect of specialization is to specialize to be different sizes. And there's a huge range of sizes in our cells. So what do you think? Which one do you think is smallest in this list of cells here? Which one do you think is biggest? Well, here's the answer. So um, red blood cells are some of the smallest cells. You can see them there, 8 micrometers. I guess that's the diameter. White blood cells, also quite small. Sperm cells, even though they're small, they're quite long because they're tails. Egg cells, I was saying, are quite big. You can see them sometimes. And nerve cells are long, so that's why they're really variable. You can have really long ones. And muscle cells can be really long. So there's a diversity in size. But in general, cells are limited in size. They can't get too big because of this issue of surface area to volume ratio. So this is a little bit of math here, but basically, if you think mathematically, so surface area is always something squared, right? And volume is something cubed. And so because of that, as a three-dimensional object gets bigger, its surface area gets bigger, of course, its volume also gets bigger, but the volume gets bigger faster because volume is cubed. Surface area gets bigger slower. So therefore, the ratio of surface area to volume, like if you're expressing surface area over volume as a fraction, gets smaller as the cell gets bigger. That's the math. Now, why does that matter? Well, there's the math again. Okay, you can see if the x is the length of one side, sort of representing the size of the cell, and you can see the red line is the surface area and the volume is the blue line, the volume's getting bigger much quicker. So surface area is important because the surface of the cell is essentially the plasma membrane. It's the place where materials are exchanged. It's the place where nutrients come in. Think of oxygen, think of glucose. Those all have to cross in through the plasma membrane, through that surface. And it's the place where wastes are released. Think of like carbon dioxide. It has to pass through the plasma membrane to get out. It's also the place where heat is exchanged with the surrounding environment so the cell doesn't overheat. Processes like cell respiration make a lot of heat. And so cells need to exchange that heat through the membrane. So that all depends on the surface area. If you have a greater surface area, you can have more exchange. But the volume determines how much exchange is needed. If you have more volume, you need more nutrients, right? You need more oxygen, you need more glucose. If you have more volume, you're making more waste. You're also making more heat, right? And remember, volume is increasing faster. So the need will increase faster than the capacity. And there will come a point where the need is too high for the capacity to meet that need. So cells have a limitation in how big they can get. Now, luckily, cells don't have to be just a cube or a circle or a sphere. They can be all kinds of shapes, and they use that as an adaptation to increase the surface area to volume ratio. So whenever you see something that's increasing surface area to volume ratio, it's allowing a cell to meet its needs without getting bigger. And so some examples. In this cell, if you look at the top, you can see it has these microvilli. It's also called a brush border. Basically, several infoldings of the plasma membrane. What does that do? That increases surface area without increasing volume so much. And so that allows for a lot more movement across that membrane. You will see this in any kind of cell which requires a lot of movement across the membrane. Now, in this example, we're looking at a proximal convoluted tubule cell. Again, a topic we'll look at again later, but a proximal convoluted tubule cell is found in the kidneys. The kidneys are the place where toxins are removed from the blood, and also water is water balance is regulated, so urine is formed in the kidneys. But basically how they work is blood enters the kidneys at a high pressure. A lot of the blood plasma, like the liquid part of the blood, gets forced into the kidneys. And then what the kidneys do is they reabsorb back into the blood anything that's needed. So like glucose goes back into the blood. Ions, sodium, and so on go back into the blood. And then whatever's left over is not needed, and that enters the urine. So there's a lot of absorption back into the blood in the kidneys. And so therefore the cells within this part of the kidney called the proximal convoluted tubule have these microvilli to facilitate a lot more absorption than would be otherwise possible. And so you'll see that in other places too, like in our intestines, our small intestine. The cells that line the small intestine have these microvilli that increase surface area so we can absorb the nutrients from our food. You'll see, like looking at a plant example, like a root hair extension, that increases surface area so we can absorb water. Red blood cells, they do something different. They form this flattened shape, like this kind of shape with an indent in it. And that, again, increases the surface area compared relative to the volume to allow a lot more oxygen to cross through the membrane. And then the oxygen is carried by the hemoglobin protein. You should remember that now. The hemoglobin protein, which is in those red blood cells. All right. Continuing our run through of examples of specialization. Next, we're going to go to the lungs. The lungs are the site of gas exchange, the site where oxygen enters the blood and the site where CO2 leaves the blood. And all of that gas exchange happens deep in your lungs in these little, like balloon-like structures, they're small, called alveoli. And the alveoli are a structure, a tissue made of two different cell types, and they're called pneumocytes. So pneumo is referring to lungs, and cyte always means cell. So we have these type 1 and these type 2 pneumocytes, and they're quite different, and they have different roles within the alveoli. So there's a nice diagram of the lungs, and you can see the lungs, you have your trachea here, and then it branches into two bronchi, and then the bronchi branch off into all these little bronchioles, and then at the very ends of the bronchioles are these alveoli, these little sacs where the air will enter when you breathe in. And the alveoli are covered in capillaries which carry the blood. And then what has to happen is the oxygen has to travel across the cells of the alveoli into the blood, and the carbon dioxide has to go the other way. There's a nice diagram showing the place where an alveoli, alveolus, one alveolus, and a capillary, a blood vessel, are meeting. And you can see where the oxygen and the CO2 are moving. So there's two kinds of cells. There's type 1 and type 2. So most of the cells of the alveoli are these type 1 cells, and they're very, very thin. So how does that structure facilitate its function? Well, it decreases the distance for the gases to travel across those cells. There's also these type 2 cells. They are not flat, so their function is not gas exchange. But what they do is they release a liquid called surfactant. And what the surfactant does, it's sort of like detergent almost, is it prevents the surface tension that's formed whenever there's water present from causing those alveoli to sort of collapse and stick together. The alveoli are these very delicate structures. They're just a single cell layer thick. It's like a plastic bag, but thinner. So imagine you have a plastic bag, but thinner, and it's moist in there. What's going to happen when the sides of the plastic bag touch each other? They're going to get stuck, right? That's a problem, because we need them to be like open so air can come in and go out. And so this surfactant made by the type 2 cells prevents the alveoli from sort of sticking together and collapsing on themselves. And here's a way better diagram showing the different cell types. If you look So on your left, you can see an alveolus surrounded by blood vessels, and you can see in the blood vessels you can see some red blood cells. Okay, it is a little bit difficult to orientate yourself to these diagrams. Then you can see the type 1 pneumocytes and type 2, and notice the type 1 are flat and type 2 are not flat. Okay, in the diagram on your right, you can see like a more clear drawing of that. And you can see there the type 1 very flat, a type 2 releasing surfactant prevents the alveoli from collapsing. There are also macrophages in your alveoli. Those are those immune system cells that do phagocytosis to destroy any invading pathogens that might be in the air you breathe in. Okay, we're getting close. Moving on to muscle. Now, muscle cells are very unique. And there's different kinds of muscle cells. We have muscle cells that are attached to our bones, which allow us to move. Those are skeletal muscle cells. Those are the ones you probably think of most often. They're quite unique because they're very long, and they actually have multiple nuclei per cell. So it's like many different cells almost fused together to make this long cell with many nuclei. So they're quite unique because usually we consider a cell to just have one nucleus. Skeletal muscle cells have more than one nucleus. The other kinds of muscle are smooth muscle. That's the muscle that your organs are made of, like, for example, your small intestine, your esophagus, and so on. So not attached to bones, but they can contract your stomach as well to achieve their function. Cardiac muscle is very unique because it's the muscle of your heart. It's the muscle your heart is made of. And your heart is extremely important. If it stops beating for too long, you're going to die. So the cardiac muscle is unique in this ability that it functions for your whole life and is very effective. Its structure is quite different. So looking at cardiac muscle compared to skeletal muscle, cardiac muscle is branched instead of, and skeletal is not. Cardiac muscle has only one nucleus per cell, whereas skeletal muscle will have multiple. Cardiac muscle, the individual cells are connected to each other in a very special way. Like, it's almost like this kind of connection. It's like a strong connection called an intercalated disc. And that makes sure they can almost act as a single unit. And they're quite short, much shorter than skeletal muscle. The branching, the connections, the shortness, all allow cardiac muscle to work together more effectively and beat in a coordinated way. So think of your heart. It has to beat in a very coordinated fashion. The atria, you don't need to know the parts of the heart yet, but one part of the heart has to pump first and get the blood to go down into the ventricles, and then the ventricles have to contract and push the blood out to the lungs and the body, and that has to happen in a coordinated way. And that's only possible because these cells that form the heart are connected in a very specific way. So it's only possible because of their unique structure. Okay, we're nearing the last two slides. Adaptations of sperm and egg. These are gametes, is what they're called. They're haploid cells. They have half the genetic information, and when they fuse, they make a zygote, which is the first cell that forms a multicellular organism. They're highly specialized. Sperm is very small. It is long, though. It's small in width, though. They're very specialized because they have this flagellum that allows it to move. And to move that flagellum, it needs a lot of energy. So they have all these mitochondria in the middle part, the midpiece. The mitochondria are doing cell respiration, making ATP so that the flagella can move. And then it has this sort of like streamlined head so it can swim quickly. And they have a very specialized vesicle called an acrosome at the front, which releases an enzyme that helps to break through the outer layer of the egg cell and get into the egg. Excuse me. The egg cell is very large. It's not motile. It's a big spherical structure. And it basically contains all the nutrition necessary to initiate embryo development in the very early stages. I think that's it for now. We will come back to sperm and egg cells when we study reproduction again in the future. So, congratulations, you made it to the end. I think this is the longest video I will ever make. I hope you got through it. I hope it was helpful. And again, I want to emphasize, you do not need to know everything that was referred to in this video yet. This was like an introduction to many different topics to get at the idea of what organelles are, why they're important, and how cells become more specialized over the course of development, going from stem cells to differentiated cells. I hope this was useful. Thank you. Bye-bye.